Thank you for tuning into my channel. I'm Chris, and today I'm going to be discussing a paper today that I read in my nutrition class, and the paper is Ranking Possible Carcinogenic Hazards, and this is written by Dr. Bruce Ames, and his picture is right here. So Dr. Bruce Ames is a biochemist, and he's a professor at Cal. Whoop. And he was awarded the Mott Prize in 1983, and the paper that I'm going to be discussing is a review paper that he wrote in 1987. As the first sentence of the review states, animal testing cannot be used to accurately predict human risk. And the way this paper does this is by using and developing this value called the Herp value, H-E-R-P. Herp stands for human exposure rodent potency, and it's given as a percentage point. The Herp value uses two different indices. The first index is a human exposure index, and it shows how often or at what concentration a human is exposed to a certain carcinogen or a certain chemical. The second index is the Carcinogenic Potency database, and it's abbreviated as CPD, and this uses TD50 values for development of a tumor. A TD50 value halves the likelihood that a mouse will end a trial being tumor-free. So you have two groups of mice when you're doing a TD50 experiment. One's a control, one's a variable. And in the control group, you're going to go and track them through their entire life, 100 weeks. And maybe out of the 10 mice that you have in the study, only 2 of them develop tumors. So 8 healthy mice, 8 tumor-free mice exit the study. Now you look at the variable group. Now the control group had 2 tumors, but 8 healthy mice. So the TD50 actually halves the likelihood that the mouse will exit a trial tumor-free. So that out of half of 8 is 4. So that means that the TD50 value of a chemical in this experiment would be 6. So whatever that the concentration given, or the milligrams of that chemical given to that mouse, would end up yielding 6 mice having tumors within their lifetime. So they use TD50 value, and this uses like rats and mice and different species of both, and they cherry pick the one that has the lowest TD value, and the lowest TD value means the highest Herp value. Going back into the paper, the first thing that Dr. Ames addresses in the paper is the comparison between man-made pesticides and natural pesticides. Plants have been making their own pesticides to fend off insects, and humans came along recently and we decided to make our own pesticides to increase crop yield and such. He analyzed the DDT for human exposure had a 0.0003 value, and then what he wanted to do is he wanted to compare against plant-based pesticides, ones that the plants make themselves. And first he stated that by weight, we consume 10,000 times more plant pesticide than man-made pesticide. But this doesn't tell anything about the potency. The potency comes with a Herp value. And with DDT, it was a 0.0003. And when you look at some of the plant-based pesticides, it's pretty interesting. Hydrazine, that's found in mushrooms, was a value of 0.1. Safrole, which is found in natural root beer, was a 0.2. And comfrey pepsin is a 6.2%. So the idea that, the notion that within society that, oh, it's a man-made pesticide makes it bad, is not necessarily true when you look at a Herp value. So moving on past plant pesticides, he analyzed carcinogens found in food as well. So Aflatoxin is a compound found within peanuts, and the Herp value was shown to be 0.003, 10 times lower than what the FDA says is okay. But this compared to beer, wine, and home air. Beer was a 2.8, wine was a 4.7, and just being in a house for 14 hours out of a day was a 0.6. So it kind of puts in a lot of perspective about what we think of a Herp value, and Dr. Ames tries and brings attention to the fact that many more things can be carcinogenic, referring to Herp value, than we think originally. But using aflatoxin actually was an important way to expose some of the bias that's actually found in Herp values, because Herp values use the rat species or mouse species that has the lowest TD value per compound. So they didn't stick to one species of mouse or one species of rat for all their analysis. And when they looked at a little bit less than the 400 compounds that they analyzed in this entire study, of the 392 chemicals they tested, 226 were carcinogenic in either rats or mice. But out of those 226, 96 of those, if they were positive in mice, they were negative for carcinogenic effects in rats, or vice versa. Rats and mice are a very similar species, and with the small sample of chemicals that they used, there is very low correlation between carcinogenic hazard of a certain chemical. So comparing that from mice to humans, or rats to humans, is even further of a jump and further discredits the ability to use animal models to determine the hazard of a chemical with humans. And as I said earlier in the video, they did a standard life analysis for the TD50, which was like 100 weeks. I mean, they did it a little bit shorter because you get more tumors through aging. The life expectancy of a human and a mouse or a human and a rodent is a lot different, so there can be more complications on that front. Furthermore, more issues with bias is that the possibility of synergistic effect or destructive effect. So you give a rodent aflatoxin and it has a Herp value of whatever, but humans don't just eat peanut butter all day. If you did, that's fine, but they don't always eat peanut butter, and they eat other compounds. And these compounds can have synergistic effects. Maybe you also have high salt diet and you like to eat peanut butter. Maybe there's a compound effect on this and it actually increases the Herp value of it. Maybe it has a destructive effect and like negates the effects of, like, maybe you drink a lot of vitamin C and this helps like recharge the tocopherol, so like you don't have to worry about it as much. So like there's a lot of complications in like this effect in determining if something is carcinogenic in the effect of humans versus rodents. So to summarize Dr. Ames's 1987 review paper, he really just wants to point out that it's not accurate to directly look at the rodent analysis and confer what that means for human populations. Just because something is a synthetic molecule, like a synthetic pesticide, does not necessarily mean it's worse than a natural pesticide. And he's not saying like don't eat the mushrooms or don't drink a glass of beer. He's just saying be more skeptical when stuff comes out regarding this thing causes cancer. So thank you for tuning in and watching this semi-long video. I enjoyed the paper a whole lot. It was a really good read. Thank you, Dr. Crawford, for bringing it up in class, and I'll see you guys soon. Thanks.